Hello students, welcome to the EPG Pathshala program. I am Dr. Shoaib Lukman from the Council of Scientific and Industrial Research, Central Institute of Medicinal and Aromatic Plants, Lucknow. Today I am going to talk about the properties of lipids from the paper Lipid Metabolism. Properties of lipids include to understand what property lipids possess, from these properties what they do and how they perform their functions based on these properties. Like I said before, lipids are dissimilar group of biological compounds, but they are poised of a chain of hydrocarbons ending with groups of bonded oxygen and hydrogen, and share the general property of being at least partially not water soluble. Lipids are essential to all forms of life and are the key constituent of every cell membrane present on Earth. Some of the key properties of lipids include energy storage for animals as well as for other organisms, while other properties are described in this presentation. The properties of lipids include the solubility, the hydrolysis, saponification, hydrogenation, emulsification, rancidity, and consistency. Each and every property of lipids depicts the credibility of lipids. The prime property of lipid is the solubility. As we are aware of the fact that lipids are insoluble in water, or in other words we can say that lipids are insoluble in polar solvent. But on the other hand, lipids are soluble in chloroform, benzene, ether, and other non-polar solvents. The picture shown below is showing how we can test the solubility of the lipids. There are three pairs of the test tubes. In the first pair, the lipids are mixed with water, and in the second, they are mixed with the petroleum ether. Here, oils are used as an example of lipids. As we can see after mixing, it is evident that the first tube, which is having water, the oils do not solubilize. Whereas in test tube 2, which contains the petroleum ether, oil gets solubilized. So it shows a clear depiction that non-polar mixes with non-polar and polar mixes with polar. The graph between the polar solvent and the non-polar solvent also show the length of the carbon chain present in the lipids and the solubility correlation. From the graph, it is evident that those lipids which are dissolved in polar solvent, the solubility index is poor as compared to those which are dissolved in non-polar solvents. Consistency of the lipids is another important property of lipids. Lipids, if you can see, they are colorless. You cannot smell, as lipids are odorless, and also they are tasteless. Being the lighter than water, lipids have a specific gravity of 0.86, and the consistency of lipids depends on the presence of the saturated and or unsaturated fatty acids. The picture clearly depicts the lipids, which is seen as colorless and probably lighter than water. Hydrolysis is another important property of lipids. The hydrolysis is brought about either in the presence of acids or alkalis under the activity of enzyme lipases. The picture is clearly showing that three water molecules are forced into the oxygen bridge, as a result of which the three bonds are broken, leaving a glycerol molecule and three fatty acid chain. The hydrolysis of the lipids or the fats may be the acidic hydrolysis or the alkaline hydrolysis. The acid hydrolysis results in the formation of glycerol and long chain fatty acid, whereas the alkaline hydrolysis of fats or lipids results in the formation of sodium or potassium salts of fatty acids, which is also called as soaps. And the process or the phenomena is termed as saponification. The animated version shown here clearly depicts that fat or the free fatty acid, when reacted with a base, it results in the formation of soap plus the release of water. Hydrogenation is another important property of lipids. In this phenomena, the conversion of liquid vegetable oil into solid vegetable ghee by reaction with hydrogen gas takes place in the presence of catalyst nickel at a very high temperature of 200 degrees Celsius. The pictorial representation show that polyunsaturated fats undergo hydrogenation, as a result of which there is a cleavage of double bonds and it forms the hydrogenated fats. Now there is a three different state of lipids as far as hydrogenation is concerned. One state where lipids is in natural state, another state which is in a partially hydrogenated state, and finally in a fully hydrogenated state. Emulsification is another important property of lipids, and this property takes place when fats or oils are rubbed with water. The large molecules of lipids broken down into smaller molecules resulted in the formation of emulsion. The entire phenomena or the process is called as emulsification process. In this pictorial diagram, this emulsification process is shown. We can see how fat droplets slowly added in a water phase and dispersed, finally broken down into small particles. Rancidity is one of the important property of lipids. It has been observed that when lipids or fats are exposed to heat, light, air or moisture for more than a month, there is an unusual and undesirable odor is developed. Such type of lipids which undergoes this type of phenomena are referred to as rancid lipids, and the process is called as rancidity. Rancidity may be of two types: hydrolytic rancidity or oxidative rancidity. And between these two, there are some common features like a foul odor and flavor is present in both hydrolytic as well as in oxidative rancidity. Hydrolytic rancidity always uses enzymes, but even though oxidative rancidity does not always use enzyme, lipoxygenase can also produce unpleasant flavor during oxidative rancidity. As far as hydrolytic rancidity is concerned, it is the process of breaking down a lipid or a fat and producing the glycerol as well as the fatty acids. The hydrolytic rancidity occurs mainly due to the action of enzymes called lipases. These enzymes, primarily secreted by bacteria, is also present in the gastrointestinal part of the human body. Thus, they produces a problem in dairy products. There are shorter chain of fatty acids which produces the unusual flavors, and also the heat and moisture can hasten the hydrolytic rancidity. On the other hand, the oxidative rancidity, it is the process of oxidizing fatty acid chain. There is a carbon-carbon double bond in unsaturated fats that are half broken, thus making it a carbon-carbon single bond and one bond with an oxygen molecule. And the presence of oxygen itself results in the formation of undesirable odor as well as flavor, which is a part of oxidative rancidity. The process of oxidative rancidity can be catalyzed by the light, the enzyme, the trace metals, as well as the free radical species. The physical properties of lipids include the fatty acids, which are set up as stacked layer of molecules. The thickness of each layer of two extended molecules arranged in such a way that the water-loving, the hydrophilic carboxylic acid groups shape the two faces, and the water-fearing, the hydrophobic chains outline the inner core. In case of specific fatty acids, the molecules packing might differ in forming different crystals, and the configuration is termed as polymorphs. The saturated fatty acids of biological significance have melting temperature exceeding 27 degrees Celsius or else 81 degree Fahrenheit, and this rise with escalating hydrocarbon chain lengths. Both the polysaturated as well as monounsaturated molecules liquefy at considerably lower temperatures than their saturated moiety. In most of the biological molecules, the melting temperature becomes lowest when the carbon-carbon double bond are positioned in a close proximity to the center of the hydrocarbon chain. At room temperature, these molecules form viscous liquid, and the hydrophobic nature of the majority of fatty acids surpasses the hydrophilic character of the carboxylic acid, creating an aqueous solubility of these molecules very squat. At 25 degrees Celsius, the solubility of fatty acid is 3 into 10 raised to the power minus 6 per gram of solution, and the aqueous solubility lessen rampantly with the accumulation of each carbon units adjacent to the hydrocarbon chain. This association imitates the energy necessary to transmit the molecules to water, forming a pure hydrocarbon solvent. With every CH2 group, for example, additional energy is obligatory to organize water molecule of fatty acid in the region of hydrocarbon chain, resulting in the hydrophobic effect. The carboxylate group separates a positively charged hydrogen ion in pure water to a very smaller degree, thus representing the equation R-COOH, which converts or dissociates into RCOA negative plus hydrogen positive, where the R signify the hydrocarbon chain. The negative charge bearing the carboxylate ion is more polar than the undissociated acid. RCOH group can be changed to RCOA negative with the addition of equal number of base molecules. For example, sodium hydroxide. In fact, it reinstates the sodium ions with hydrogen ions to provide the fatty acid salt, which is commonly called as soap. The anion RCO anion in water impulsively shape stable spherical aggregates known as micelles that serve as useful detergent property of soaps. The micelles diameter is more or less twice the length of unmitigated fatty acid. The concentrated preparation of the micelles water dispersion display an immense cleaning power. Usually look like pure water, these dispersions are quite stable. Foams and bubbles on the exterior surface of soap dispersions results due to spontaneous adsorption of RCO negative ions at the edge between the air as well as aqueous dispersion with the mechanically stretched, vigorously stabilized effect between the air as well as water interface. Another important property of lipids are ozonolysis. The unsaturated fatty acids are vulnerable to dilapidation by ozone. This reaction is accomplished in the acid from oleic acid. If you can see the figure, you can find out that there are oxidative workup taking place during the ozonolysis process, which results in the vulnerability of the lipid molecules or the fatty acid molecules to form several acid or esters that can be utilized for further biological processes. Coming to the chemical properties of lipids. Chemically, the acidic carboxyl group of the fatty acid is the most reactive portion. It reacts with alcohol to yield esters, releasing the water molecules. As well as in complex lipids, the ester bond links fatty acid moieties to other groups. Ether bond also links fatty acids and are chemically more stable as compared to ester bonds. In fatty acids, the hydrocarbon part is relatively defined to chemical assay, except the carbon-carbon double bond and different molecules respond with similar double bond. For instance, when a platinum is present as a catalyst, hydrogen append to the double bond to bestow a saturated fatty acid. Halogens, including iodine, chlorine and bromine and their derivatives, for example hydroiodic acid, also retort with the double bond to structure the saturated fatty acids. But herein the halogen, one or two atoms, swap the hydrogen atoms usually endowed in the saturated acyl chain. Carbon-carbon double bond can also respond with oxygen in either enzymatically catalyzed oxidation reactions or non-enzymatic processes. The procedure engenders a range of products, several of which adds to the stale smell in vegetable products and spoiled meat. Universally, it is known that highly unsaturated fatty acid is more easily oxidized. Thus, the properties of lipids explain its consistency, solubility, hydrolysis, saponification, emulsification, and rancidity. Hardening is also an important property of lipids, since the saturated fatty acids are having superior melting point than the unsaturated ones. The procedure is called hardening. This knowledge and technology is used to change vegetable oils to make margarine. During the partial hydrogenation, the isomerization of unsaturated fatty acid from cis to trans configuration take place. Use of high temperature and high pressure forms the fatty alcohols from the fatty acids. However, fatty acid esters converts to fatty alcohols more simply. Certain unsaturated fatty acids are cleaved in molten alkali in the Warren-Trap reactions. In this figure, we can see that the unsaturated oils, when acts on by hydrogen or by heat in presence of nickel catalyst, it results in the formation of saturated fats. And this phenomena refers as the hardening of the fats or the lipids. So, apart from the above mentioned properties, auto-oxidation is also a property of lipid where a chemical change is endured by unsaturated fatty acids. The process gains momentum by the charisma of trace metals and require oxygen. Oils of vegetable origin defy this practice as they restrain antioxidants, for example tocopherol. Oils and fats and oils are repeatedly treated with citric acid, which is a chelating agent, to eliminate the metal catalyst. As far as the analysis is concerned, chemically, the methyl esters of fatty acids are alienated by gas chromatography, and furthermore, the unsaturated isomers separated through thin layer chromatography. Some of the well-known examples of lipids are described as under, as we have explained it in the prologue section. Fatty acids, for example, arachidonic acid, linoleic acid, oleic acid, and palmitoleic acid. The examples of fats and oils include the animal fats, butter, the coconut oil, the corn, the human fat, the herring oil, the lard, palm, peanut, and sunflower. The waxes category of lipids include beeswax, carnauba wax, and spermactide. The phospholipids include cephalins, lecithins, plasmalogens, phosphatidylinositols, and sphingomyelins. Glycolipids include kerosene, nervone, oxynervone, phrenosine. The steroids include C21, C24, C27, C28, and C29 steroids. The terpenes include the monoterpenes, which contains of the isoprene units, the diterpenes, the sesquiterpenes, and the triterpenes. And the last, carotenoids. It includes carotenes, lycopenes, and xanthophylls. So, today in this lecture, we have learned about the properties of the lipids, the physical properties, which includes several specialized things about lipids, and the chemical properties of lipids, which clearly speculate the incredible nature of lipids, the hydrolytic properties of lipids, which yields not only glycerol as well as fatty acid, so that they can get converted into other products useful for the cell metabolism. The saponifiable and the emulsifiable nature of the lipids, which is an important property as far as the emulsification process as well as saponification process is concerned. The rancidity, the peculiarity of the lipids through which we can understand the degradation, the oxidation of the vegetable products as well as meat takes place. The reactions of lipids as well as fatty acids are some of the well-known examples of the lipids which constitute the important property of lipids. So in this way, today we have learned the properties of lipids, which is an incredible feature responsible for lipid metabolism. Thank you.